afternoon, sir.
Okay, students, that you have all gathered. Okay, so Adhyasa Dubey, good afternoon. Okay, all students, that I congratulate to you all, and that your monthly test 4 will be held on that of 29. It will start on 29, and the bio test is on 31st. Okay, I have given the portion that you might have checked WhatsApp, and found that I have given that of biomolecules and cell cycle. Okay, you should have to read these 2 chapters, whatever I have taught in that class. All these questions are made from that of what I have taught in the classes. Okay, so you should have to read it mindfully and to answer all questions. And the pattern is same what is previously that was held in that of monthly test 2, 3. That same questions and the same type of same pattern will have to find in question. So there is no different in question pattern. Okay, students, that yesterday that we had finished that of meiosis, the first part of meiosis 1. But up to that prophase 1 we had finished. Okay, and the meiosis, that you might have known that the significance of meiosis is that to make that of that is, it is found in that of that is sexual reproducing organisms in diploid cells, and it make that of diploid cells to that of haploid. Okay, and that haploids meet together, and that again come back to that of diploid state. So diploid to haploid and again to diploid. That is the main basis of that of meiosis 1. And that meiosis that it has been divided into 2 types. One is that of meiosis 1, another one is meiosis 2. And that meiosis 1, students, you will know that it is that of reductional division, in which that means diploid become to that haploid 2n becomes ends, and but in between that of 2, that is meiosis 1 and meiosis 2, there is a gap. That gap is called interkinesis, and during the interkinesis, that duplication, centriole number of centriole doubles, and the duplication of centrioles happen in interkinesis, but there is no DNA synthesis. Okay, no DNA synthesis happen in interkinesis, and see that what happens here, when thing you see that I'm just drawing, you see that in that you see that it is a chromosomes, it is another pair of chromosomes. Okay, it is a homologous chromosome. You see that it is a homologous chromosomes, and these chromosomes, you see that this is the chromosome number 1, this is the chromosome number 2. Okay, in meiosis 1, what it happens that you know that is a homologous chromosomes, and that is they are the bivalence, that is bivalence, and that bivalence chromosomes, and these chromosomes have been made the pair. You see that, and this is the point of recombination nodules where crossing over takes place. Crossing over means that exchange of genetic material. Okay, that is, and this looks like that of except, so it is also called that of chiasmata. Yesterday I had all something discussed, and you might have understood clearly. And you see that in meiosis 1, what it happens that these chromosomes, this is a paired chromosomes, that is called bivalence or tetrad also, because there are 4 chromatids. Here is the chromatid 1, chromatid 2, chromatid 3, chromatid 4. So crossing over is that of that is crossing over takes place in between that of 2 non-sister chromatid. Yesterday I had told you that is 1, 2 are sister chromatid, but that is 4 and 3 are, that is they are also sister chromatid. Okay, sister chromatid, but that 2 and 3, you see that 2 and 3 are sister chromatid. Okay, that is non-sister chromatids. So meiosis 1's objective is to divide okay, to divide that chromosomes, that bivalent chromosomes into two. Okay, one will be like this, and another one will be like this. Okay, that means how to separate them. That means separation of a bivalent chromosomes into two units is the objective of meiosis one. Okay, objective of meiosis one is to separate two homologous chromosomes. Is to separate two homologous chromosomes which are attached to each other. Okay, which are attached to each other, and to make them two chromosomes. Make them two chromosomes. But what meiosis 2 does? That is meiosis 2. Students, you see that mindfully. Meiosis 2, that means break this. It break these chromosomes into two parts. Into two parts. You see that. These are the, this is a chromatid, this is another chromatid, this is a chromatid, this is another chromatid. To break the two chromatids into two separate units. So you see that. That means here is a chromatid, here another chromatid, here is a chromatid, here is another chromatid. So in that, you see that there are two chromosomes. Two chromosomes having four chromatids. So that will be separated in meiosis 2. So meiosis 1 separates what? Separates homologous chromosomes into two units, And meiosis 2 divides these two chromosomes into four parts. And that four parts also what? These are the chromatids that are condensed to chromatin, and that makes a nucleus. So there are four nucleus, so there are four cells. Okay. So the meiosis 1 objective is to divide the homologous chromosomes into two units. And then meiosis 2 objective is to divide each chromosome into two units, two chromatids, into two units. As two chromosomes are there, so there will be four chromatids. Each chromatid will convert into, that means they will be decondensed and will form that of a chromatin. And that chromatin, and there are four chromatins that will make that of four nuclei. Okay, so there will be four cells. That is the goal of that of meiosis 2. Now you will have to elaborately discuss. Yesterday I had told you that of prophase 1. Students, have you remembered that of in prophase 1 it is the longest phase of that of meiosis 1. In that of prophase 1 there are 5 subphases. Have you remembered that of 5 subphases, or you have forgotten? One by one.
Remember, sir.
Sir, remember.
Leptotin. Leptotin.
Leptotin. Second one,
Zygotin.
zygotin. Third one,
Passion.
acetin. Fourth one,
Diplotin.
diplotin. And the fifth one is
Dikinesis.
dikinesis. Very good, brilliant students. Okay, I'm bringing duster. Okay, you see that. That means what it happens that I have told you that meiosis 1 and meiosis 2. Then I will elaborate, we discuss today, and I will finish this chapter. Okay, and you see that. First of all, in that first is leptotin. In the leptotin, what happens that compaction of chromosomes. Okay, compaction of chromosomes. That is main events. Chromosomes. Then you see that in that of after leptotin, that is zygotin. In zygotin, what happens that means synapsis. Okay, that synapsis. Synapsis, bivalent, or that of tetrads. Okay, tetrads. That is homologous chromosomes. Formation of homologous chromosomes. Okay, these events. In that of pachitin, that recombination nodules are formed. Okay, recombination nodules are formed. And recombination nodules, and it was helped by that of a recombinase, that is enzymes, and also another two enzymes also support that of two recombination nodule for crossing over. Have you remembered other two enzymes? One is recombinase. There are three in your NCERT books, that is recombinase enzyme given. But yesterday I had told there are two other enzymes also helpful in that of crossing over. Which
Sir,
other?
endonuclease and ligase.
Yes, very good. Endonuclease and ligase. Brilliant students. Okay. So that recombination nodules, that formation of recombination nodules where crossing over takes place in recombination nodules. And that crossing over is being, that means it is held by that of recombination enzymes, endonuclease and ligase. And pachitin and in diplotin. So what you will have to find out that diplotin, that is Guy's matter. Okay, that is Guy's matter. Guy's matter. That is except. Okay, except that is these are that of two chromosomes. You see that this chromatid will have to make that of a except structure. So that is called Guy's matter. And diplotin, and in that of dikinases, in that of dikinases, that is dissolution of Guy's matter. That is dissolution of Guy's matter. Guy's matter, and that of then dissolution of Guy's matter occur in that of diplotin. Okay, diplotins, and that then that is a reappearance of that of that is called what? That is DNA. That is nuclear membrane. So all these things happen in that of dikinases. So these are, yesterday I had a discussion now, lot of things. I'll send notes because I am completing this chapter today. So after the end of this class, I'll have to send the notes what I have not sent yesterday. Okay. So you see that today now you are going to discuss about that prophase 1 you had finished. Now you see that of metaphase 1. Okay. Metaphase 1. Students, it is very important that you have to mark it. That is metaphase 1. Okay, metaphase 1. You see that in that of metaphase 1, what we'll have to see? Mainly you see that, suppose this is a cell. And in that cell, in metaphase 1, what happens that students, you see that that means all chromosomes, chromosome means that bivalent chromosomes, okay? That bivalent chromosomes will attach to that of that is equator. They will come to that of center. So you see that I am drawing that. Okay, you see that one I am drawing that. You see that this one, another one I am drawing. Okay, another one I am drawing, and you see that these are, and another one I am just drawing. You see that. Okay, this one. These are the bivalent chromosomes, bivalent, because they are paired chromosomes. Okay, another I'm just drawing that one. You see that I am drawing this one. Okay, I have drawn here three chromosomes, and they are in the middle. They are in the center. You remember that in metaphase of mitosis, what I had told you that means there are chromosomes. These are the chromosomes. Okay. These are the chromosomes. You can have to find that in such chromosomes, what you will have to find? You will have to find that here are the one kinetochore, here is another. Here is one, here is one. Here is one, here is one. Okay. And here that centriole, that you see that is spindle fiber, that spindle fiber join, and that spindle fiber join like this. Okay, but here one chromosome having two kinetochores. Okay, one chromosome in that of metaphase of meiosis. In that metaphase of mitosis, there are two kinetochores. Okay, two kinetochores in one chromosome. Okay, in one chromosome. Here you can see that there are paired structure, there are homologous chromosomes in that of metaphase 1 of meiosis. In metaphase of mitosis, two kinetochores are found in one chromosome. You see that here is a chromosome, here is a one kinetochore, another one. Here is one, and that is one chromosome having two kinetochores. But here you see that there are paired chromosomes. There are two chromosomes. They are two chromosomes, but they are Bivalents, they are homologous, but they have also two kinetochores. They have also two kinetochores. So understood that in case of mitosis, one chromosome has two kinetochores. But here in case of that of metaphase one of meiosis, two chromosomes have only two kinetochores instead of four. Okay, as they are bivalent, they have two kinetochores. So here is that of, you see that here are that of centrioles. So these are that of microtubules. These are the microtubules, and these microtubules, you see that, and the one will that you see that one thread, that is fiber, that is spindle fiber, join this one. Another fiber coming from the up, then bind this one. Here also something happen. This one will come from this one, will come bind this one. This one will go this one. From this one will bind this one. This one will go this one. Okay, like this. But they are attached. They are attached here. Okay, that is they are attached here because they are homologous chromosomes and they are synapse. That you see that means they are bivalent. Here they have attached to each other. They have attached to each other. Okay, attached to each other. So you see that. That means this one. That means this one. That means this one drawing this one. That means it has pulled it, and it is also bind to this like this. So like this way. So you see that. That means this one is, and this one is like this. So you see that this one has, that means this one has taken like this way, and this one has taken like this way. Okay, so that you see that. But in this case, you see that in this case, that is one, that is kinetochore, that is one chromosome. Each chromosome has two kinetochores. So that is spindle fiber going here and touching this point, and another also touching another kind of core. So like this way, and but here are, that means here are, you see that from this upper centriole, that is from that upper centriole, you see that one that is one spindle fiber joins one, that is kinetic core, another spindle going from that of other centriole, lower centriole attached to that of another kinetic core. So that you see that here is one, here is, suppose it is the center, I have not drawn on the center. You see that there will be one that is metaplate. Okay, that is one equatorial plate here. Here is in mitosis one equatorial plate is formed. But in case of metaphase one of meiosis, there are two equatorial plate, students. You mark that there are one equatorial plate here, another equatorial plate here. Okay, so double metaphasic plate is found in that of metaphase one. But in that of metaphase of mitosis, that is single metaphasic plate. Okay, so difference is that here you can find that of a double metaphasic plate. That is double metaphasic plate. Okay, that is also called double, that is equatorial plate. Okay, that is equatorial plate, double equatorial plate. Here also, but here is, that is single equatorial plate. That is single equatorial plate. In mitosis, single equatorial plate is formed, but in metaphase of mitosis, single equatorial plate is formed. But in case of metaphase one of meiosis one, that is a double metaphase plate is formed. You mark that is a double one. Okay, this is the double one. So double metaphasic plate is formed. That is called double equatorial plate. That is the basic difference in between that of mitosis, metaphase of mitosis, metaphase one of meiosis one. Okay, here you can because they are another, that is kinetic core. That means you see that two kinetic cores in one chromosome, but here there are two chromosomes, paired chromosomes having two kinetic cores. That is the basic difference in that of metaphase 1 of that of metaphase 1 of meiosis 1. Okay, that is the metaphase. Then we will have to go. So you have to remember that double metaphasic plate. All the chromosomes will have to come into the center. They will lie on the center on the equator. They will align to that of equator, and they will have two, that is kinetochores, each attached to that of each centrioles. Okay, that is by that of that is fiber. Okay, spindle fiber. So spindle fiber join, each spindle joins that of one kinetochores. Like this way, they are attached to each other and made a double metaphasic plate, or that is called double equatorial plate. Okay, that is the basic thing of metaphase. Same as that, they will come to that of middle, they will come to the center and form that of equatorial plate. That is the basic thing in that of metaphase 1. Okay, then come to that of then we'll have to come to that of anaphase 1. Okay, it is very important, students. You should have to mindfully watch that of anaphase. Okay, in anaphase 1, you mark it that what happens. You see that in case of that of you see that I'm just drawing that of two homologous chromosomes. They are seen as, okay, they are, that means they cross to each other. So this one is that of recombination nodules. Okay, they are paired. Okay, they are bivalent chromosomes. These are the bivalent chromosomes. Bivalent. Okay, bivalent. These are the bivalent chromosomes, and this is the recombination nodules. Okay, and that means in case of anaphase, what will, that is anaphase 1's aim is, objective is to separate these bivalent chromosomes into two separate units. So that means you see that is synaptonymal complex. That means you know that yesterday I had told you that is synaptonymal complex. That is a protein, that is a chemical that attaches two chromatids, two non-sister chromatid. That is chromatid number, that is 1. This is 2, that is 3, that is 4. So these 2 and 3 chromatids have been attached. So that will be separated. That separation is called, students, that is disjunction. Okay, that separation of non-sister chromatids is called disjunction. That term you must have to remember. That means that will be separated. Okay, that will be separated. So one will be like this, another one will be like this. So they are not attached to each other. So no more they are attached to each other. They are separated, and that separation is promoted by anaphase promoting. Okay, that's anaphase promoting factor. There is a protein, anaphase, that is anaphase promoting factor. Okay, that means this protein promotes the separation of, as you know that there is a protein that is synaptonymal complex that helps in binding of two chromatid, non-sister chromatids. That is called what? You know that is crossing over. But here is that another protein, that is anaphase promoting factor. What it does, that means it separates the two homologous chromosomes, bivalent chromosomes, into two separate units. That means bivalent chromosomes are divided into two separate units by a protein that is called anaphase promoting factor. So it is opposite of that of a synaptonymal complex. Synaptonymal complex promote the crossing over. That means attachment of non-sister chromatids. Whereas anaphase promoting factor, that means helps in separation of non-sister chromatid, which is called disjunction. So that one will go up, one will go up, another one will go down. So that separation is called segregation. Is called segregation. So when you remember that separation of non-sister chromatid is called disjunction, the process of separation, how they become separated. That is one chromosome, that is another chromosome. But in this case, you mark it that means when one chromosome will go up, another chromosome will go down. This type of placement, one will go up, one will go up, another one will go down. This is called segregation. This is called segregation. I am drawing it and showing to you. Okay, and it is very important. You will have to remember. You also draw it for your practice. Okay, for your practice students, you draw it also. That means you see that, students. That means suppose it is a cell. Okay, cell. Cells. You see that. That means here what happens that these are that of one, that is microtubules. I'm drawing it in large way. In large way, I'm drawing, students. You see that. You see that. That is one centriole, another one. Okay. You see that. That means here what it does. That means you see that. That means separated. That means you know that disjunction happened. That means how they are separated by anaphase promoting factor. So that chromosomes will, that means will look like this. You see that chromosomes will look like this. Okay, another I'm drawing like this. You see that one, another one. This one also, that you mark that. This one, another one. Okay, this one. So this one, that of in anaphase, that means the paired homologous chromosomes separated. In anaphase, that means paired homologous chromosomes become separated. And that means in that of metaphase one, they are aligned together, they are attached together. But in anaphase, that chromosome separated. Okay, that chromosome separated. And that chromosome separated, you mark it that in this anaphase, that means you know that Gregor Johannes Mendel's law of assortment. Okay, that is Gregor Johann Mendel. Okay, Mendel, you know that is father of genetics. Okay, he promotes, that means he has devised a law, that is law of independent assortment. Law of independent accessment. You see that. That means here is that of, you see that here is a chromosome. Suppose these chromosomes, first middle I have, this is the A, chromatid A. This is chromatid A. This part is that of a chromatid, suppose small a. This one is that of suppose capital B. This one is that of small b. This one you see that is capital C. This is small c. Okay. That means what law of independent assortment by Mendel, what is told that you see that. That means when they will be taken, that means when they will be separated, you see that. That means these chromatids, suppose B1 taken along with that of B, that means B, this chromatid or B. That means that will be B or that of small b. That can be taken together. Okay, that can be taken together. But B along with A also be taken. B along with A also be taken, because they are separated to each other. But there is no rule that only chromatids B or chromatid that is small b, that will be taken together. That has not been told. Okay, that means B can take also A along with it. Small a also can take B, small b along with. That is capital C also take that of small c along with it. Because you see that, because when they will have to be separated, that they can have to take anyone from them. Okay, from that of way, when they will have to be separated, they can be taken any part of that of a chromatids. Okay, they can take B, capital B small b, or capital B small a, or that of capital B small c. Okay, like this way. If here how many chromosomes are there? You see that here are the 3 chromosomes. Here are the 3 chromosomes. So 3 chromosomes. So you see that is 3 chromosomes will have, that is 8. That means a number of 8. That means you will have to find that they will have to make that of chromosomes. What they will make? That is they will have to be, that is number of combination. We will have to find that if there are 3 chromosomes, okay, that is they will make 2 to the power 3 combination. 3 to the power combination. So combination, that means what combination will make with B? That is B or that of small b, or that of small a with capital A with small a, or capital C or small c. It has not been devised. That means that chromatids can take with it, along with it, either a small a or small c or small b, or that of A with that of B or A or C. That means it is attached chromatid, it is not a factor. They can have to take that along with it any chromatids with it. So that this is law, that is 3 chromosomes. If 3 chromosomes are there, how many combination will be made? 2 to the power 3. Can you tell me that how many? That is 3 pairs of chromosomes. There are 3 pairs of chromosomes. 3 pairs. Here are 3 pairs. 1, 2, 3. That is another 3. So 6 chromosomes. How many pairs? 3 pairs of chromosomes. These 3 pairs of chromosomes will make how many combinations? 2 to the power 3. That is 8 combinations. 2 into 2 into 2, 8 combinations. Like this. Can you tell me that human beings have how many chromosome pairs?
23.
So how many combinations will be made? 23 pairs of combination. 23 pairs of chromosomes. So how many combinations will be made? 2 to the power 23. Okay, 2 to the power 23 combination. Okay, 23 combination. So owing to that reason, that means you see that there is a variation. Okay, variation happens. Variation happens. You see that genetic variation happens. Genetic variation happens found in anaphase 1, and in that of you see that in that of leptopachitin. Okay, in pachitin. That is in pachitin. Pachitin of that is recombination nodules. So genes transfer, that is genetic material transfer. So that will be recombination. So variation found in pachitin subphase of prophase 1 and anaphase 1. That is very important, students. You have to remember. Variation generally found at which subphase of prophase 1? At which subphase of prophase 1 variation is generally found? At, that is pachitin. Okay, at pachitin. That is subphase of prophase 1 in meiosis 1. In meiosis 1, that is in prophase of meiosis 1, at which subphase variation is generally found? That is pachitin. And in which phase of meiosis 1 variation is also found? That is anaphase 1. In anaphase 1 and pachitin 1, you can have to find the variation. Okay, variation. That is anaphase 1. Anaphase 1 and that is pachitin 1. That means you can have to find variation. In pachitin, what happens? Homologous genetic combination, that is recombination nodules are formed. So genes are crossing over takes place. So gene transfer, so genetic material transfer from that of one chromosomes to another chromosomes. In this way, variation takes place. So variation takes place in pachitins, that is subphase of that of prophase 1 of meiosis 1. Another phase of meiosis 1 also in anaphase 1. also that variation takes place. So that point you have to remember. And the law of independent assortment. So you should have to see that that is three pairs of chromosomes will have 2 to the power 3 combination. That means one pair of chromosomes will have 2 to the power 1 combination. So 3 pairs of combination will be 2 to the power 3 combination. 23 pairs of chromosomes will be 2 into 23. That is what? Combination 2 to the power 23 combinations. So like this way that combinations are made. Okay, combinations are made. So what law of independent assessment has told? Law of independent assortment has told that, okay, it is very important, students, that you first have to remember that segregation of one pair, that is segregation of one pair is independent to segregation of other pair. Okay, segregation of one pair, segregation of one pair is independent to the segregation of other pair. Okay, other pair. So that this one B can, that is, it is not a law that B can be taken by small b, or capital L can take only A, or capital C can take only small c. Capital B can take A also, capital B can take C also, like this. So segregation of one pair is independent to segregation of other pair. It has been told by that of law of independent assortment. Okay, law of independent assortment. What it has told by that Gregor Johann Mendel, that segregation of one pair is independent to the segregation of other pair. So anaphase based on that of law of independent assortments. Okay, assortments that you have to remember, that is anaphase. Then from that of anaphase, one will have to move to that of, so chromosome numbers half. But you have to remember that is chromosome numbers become half. So you see that means chromosome number. This one that of homologous or paired chromosomes, that chromosomes become half. That means chromosomes, that is in anaphase 1, the paired chromosomes become 1 unit each. Okay, you see that these were the paired. This pair become independent to each other. Okay, this one become one chromosome, this one is the another chromosome. That is anaphase. Then from that of anaphase, we will have to move to that of telophase. Okay, we will have to move to that of telophase. You mark it that of telophase. What happens in that of telophase? That is telophase. Okay. You see that is chromosomes. You see that is telophase. In telophase, that is very important that you will have to remember. That is a nuclear membrane and a nucleolus reappear. You can have to see that is in telophase. You can see that. Suppose you see that is, I am drawing just, you see that this one is what happens, that you see that in telophase you can have to find that nucleolus. Okay, that is nuclear membrane. Nuclear membrane which was not found earlier can be found in that of a telophase. In that telophase, you can have to find that of a nuclear membrane. So you see that means this you see that in that of anaphase 1, in anaphase 1, that means all that of you see that means these are the chromosomes. These chromosomes are being, that is, these are that of centrioles. Okay, centrioles. These are that of centrioles. So that became what? That means that were decondensed in that state. So they are being condensed in that state, and will be that is turned into the that is chromatins. Okay, in that phase they will be turned into that of chromatin. So this one will be, that means after that of they will become, so they will have to be make that of a nuclear membrane. So nuclear membrane will be formed here. You marked it that. That is nuclear membrane. So nuclear membrane formed, and this one go to that. These chromosomes become that of a decondense, and they appear in that of in that form of chromatins. Okay, in that form of chromatins they just appear in that of telophase. So nuclear membrane, these are the nuclear membranes. Okay, that is nuclear membranes. That nuclear membranes appeared here also, that of that is nuclear membranes. That is nuclear membrane appeared. In that case, another case is that of that is nucleolus also reappear. Two nucleus forms, you see that. That means two nucleus forms. These are one nucleus, another nucleus also forms. Okay, and these chromosomes become that of that is chromatins. Okay, that becomes chromatins. So that two nucleus form, there you can have to mark it that there are two nucleus. Okay, two nucleus. So nuclear membrane reappear. That is nuclear membrane reappear, and that is two nucleus form. Okay, two nucleus form. So in telophase, nuclear membrane appear and the two nucleus form. So that these chromosomes become that of decondense to form that of chromatin, and it remains in that of two nucleus. So two nucleus form in that of telophase one. Okay, and then cytokinesis form, and in that case that is called, you see that, that means it was, that is what you have marked at this one. You see that these are the bivalent. These are the bivalent tetrad. Okay, tetrad. So tetrads that are divided into two types. That is two types. That means these are called the dyads of the cells. These are called the dyads. Okay, dyads of the cells, because they are two in numbers. These are, you see that bivalent tetrad in that they are four in numbers, but here you can have to find they are two in numbers. What previously four in numbers, they now remain the two in numbers. That is called dyad of cells. You see that here is a one, another one. So these are called that of a dyad of cells. And in between that of a telophase one, in between that of meiosis one and to that of meiosis two, there is a gap. There is a gap, and that gap is called that of interkinesis. Okay, that gap is called that of interkinesis. And in that interkinesis, you see that no DNA synthesis takes place. Okay, no DNA synthesis takes place. That means centrioles here for that of you see that. Next step is that of what? That is spindle will develop. So that spindle will be, here will be what? The centrioles. That is centrioles. Centriole. Number of centriole is doubled. Okay, in that of interkinesis, number of centriole is doubled, and no DNA synthesis. Okay, no DNA synthesis takes place here. So number of centrioles is doubled in the interkinesis, because why number of centriole is doubled in the interkinesis? In between that of after telophase, that is interkinesis phase comes. This phase comes. This phase, that means for little time, it is temporary. It is temporary and momentary also. For short duration, it is a short duration. In this phase, that means number of centriole is doubled. Because why number of centriole is doubled? Because then spindle fibers will be formed. That is, they will have to what? Attach to that chromosomes. So that chromosomes to be attached. So in this case, that is spindle fibers. That is number of centrioles which are necessary for that of spindle fibers. They are formed. Their number is doubled. And another point of interkinesis is that it is a short-lived. Okay, that interkinesis is short-lived. It is a short-lived. And another point you have to remember that no DNA replication takes place, and that means another point is that, students, and this one is only centriole double, and that is have more condensed. So more condensed DNA. More condensed DNA is found in interkinesis stage. More condensed DNA. More condensed DNA is found in that of interkinesis phase. So interkinesis phase is found in between two meiosis 1 and meiosis 2. This is the first point. Second point is that this is a short-lived. Okay, that is interkinesis is short-lived. Another point is that during interkinesis, that is number of centriole doubles. Okay, number of centriole doubles. Then fourth point is that it is more condensed DNA is found in interkinesis space. It is very important. Okay, that 4 points you have to remember in that of interkinesis. That interkinesis is found in between that of two, that is two division, meiosis 1 and meiosis 2. First point. Second point, it is short-lived. Third point, number of centriole doubles. Fourth point, more condensed DNA is found. That things you have to remember. These are the features of interkinesis in between meiosis 1 and meiosis 2. Then students will have to move to that of meiosis 2. Okay, that is meiosis 2. And in meiosis 2, we'll have to find it that after telophase, that means what you have known that. That means two nucleus form in a cell, two nucleus is formed. After telophase 1, that two nucleus are found in one cell. Okay, one cells, and then we'll have to mark it that how decondensation occur of that of chromosomes. You see that. That means here is that of telophase. In that of telophase, that nuclear membrane appear. That is nuclear membrane appear, and you can have to find that of chromatins. Okay. Here also that of another nuclear membrane, so that is two nucleus, this is one nucleus, another nucleus. Okay, and that you can have to find that of chromosomes. Okay, then they are condensed. You see that they are condensed. That means a decondensation occur. When that of decondensed, it is telophase. That is telophase 1. Okay, that is telophase 1. So after decondensation, you can have to find that. Okay, what you'll have to find here? We'll have to find here that means that is chromosomes, that these chromosomes, okay, that is condensation, that in that process, that means you can have to find that is same way, you can have to find that here you can have to one centriole, another. That centriole will go to that of a polar regions, like that of mitosis, and they will have to bind, they will have to bind with, and you see that is spindle fiber. Okay, that spindle fiber, you'll have to see that of spindle fiber like this. Okay, spindle fiber. So spindle fiber forms and joins that of kinetochore. And you can have to see these things happen. You see that if you have to take this one, the larger one, you see that this one happened. That is first nucleus. You have to find the nucleus position here in a one cell, and another one also you can have to find. That is also same thing here at that of centrioles. That centrioles also. That means this one will be. That is, you see that join together. Okay, join this ones like this way. So in that that is in one cell we will have to find that of development of two nucleus. That two nucleus, you see that means two nucleus. That what happens that means there is a condensation. So after condensation, that is after condensation. That is condensation. That means chromosomes. These chromosomes are formed. These chromosomes, okay, these chromosomes are formed, and these chromosomes, that means these chromosomes are attached to that of spindle fibers. That spindle fiber is developed from that of centrioles, that is centrioles from that of centriole microtubule. These are from that of microtubule, that spindle develop and they attach to that of chromosomes. Okay, kinetic force of that of chromosomes. So that things happen in a cell in a twice manner. You see that there are two. This is the one nucleus, this is the another nucleus. That is first nucleus, this one is the second nucleus. This is the, that is first, this is the telophase 1 and this is that of prophase 2. Okay, prophase 2. You mark it that. In that of, in between that of telophase 1 and prophase 2, there is a interkinasis. That is interkinasis stage. In interkinasis stage, I have told you that that means duplication of, that means number of centriole doubles. Okay, there are two centrioles earlier in that, but here they become that of 4 centrioles. You see that 1, 2, 3, 4. In one cell, there are 4 centrioles are there, and that centrioles develop that of what? Microtubules. That microtubules join with that of chromosomes. Okay, like that of chromosomes. So that happens in that of prophase 2. So condensation of chromosome is the main important point, and that centrioles go to opposite poles. That exactly what in that of mitosis, same thing in that of prophase 2. Okay, going to that region, it is called equational divisions. So that you see that things happen. Centriole moves to opposite pole, then microtubules develop and the spindle fiber join to that of chromosomes. Okay, that means each chromosomes have 2 kinete force, and that kinete force join with that of opposite spindle fiber. That happens in prophase 2. Then come to that of metaphase 2. Okay, then we'll have to move to that of metaphase 2. This is very important, and that metaphase 2 all will come to that of that is, you see that in that of metaphase 2. That is metaphase 2. You see that this is one chromosome, that is another chromosomes. Okay, these are the chromosomes. They will come to that of the middle position, central position, to the equator. Something. But that is metaphase. At the metaphase, all chromosomes will come to that of that is equator. Okay, they will align to that of equator. So you see that that is what? That is a centriole. Here is another centriole. So they will have to be attached. Okay, they are attached. You see that they are attached to each other. Okay, they are attached, and that is what? That is called equatorial. Okay, that is called equatorial plate. That is equatorial plate, and in that case, that is equatorial plate, and they are come to that. Here later, this is the equatorial plate. Here you will have to make that up centrioles. Okay, you see that. That means one is attached, another. You see that this one, like this one. Okay, that is spindle fibers. Okay, these are the spindle fibers. This is spindle fiber. Okay, and another that this one not. So that's spindle fiber. So this one, that means this is a equatorial plate. Mark that. It is not a double phase equatorial plate like that of metaphase 1 of meiosis 1. It is not a double phase. It is a single phase. Okay, it is a single phase equatorial plate. Okay, that is equatorial plate. Equatorial plate. So you see that this is the chromat, that is that of what? Equatorial plate. This is the equatorial plate. And you mark that. That will be also, that is, you see that things happen. That things happen. I have drawn it in one, and you will have to draw it again and so two. In that of 2 cases also, that things happen. Suppose it is in one cell, it is for the development of that of that is in one nucleus, you can have to find all these things. But if you have to draw, that means if you have to draw in a large way, if you have to draw it in a large way, you can have to find also in that of second, nucleus also, that same things happens. Okay, same thing happens. You can have to mark it that. That means that is spindle fibers joining it. Spindle, these are the spindle fiber joining it. Okay, so that you can have to see that this is the equatorial plate in that of another nucleus. This is the one nucleus, this is another nucleus. So something happens in 2 nucleus. That is equatorial plate. That is equatorial formation of equatorial plate. That is the main objective of that. Okay, and the chromosomes align at the equator from equatorial plate. Microtubules form opposite poles of spindle fiber attached to each other. So microtubule spindle fiber of opposite pole attached to that of chromosomes. Okay, these are the chromosomes. The kinetochores of that of kinetochores of chromosomes attached to. There are two kinetochores. They are attached to that of that is spindles. Okay, these are the spindle fibers that mark that of spindle fibers. Okay, so so this is that of metaphase 2. Okay, that is metaphase 2. Then come to that of metaphase 2, then come to that of anaphase 2. Now we'll have to come to that of anaphase 2. In that of anaphase 2, we can have to see that in that of anaphase 2, you can have to see here. Suppose this is that of shell. Okay, and here we see that in anaphase 2, this one is that of anaphase 2. In anaphase 2, I have told you that means this is a chromatid, this is another chromatid of a chromosome. That will be separated. That means it will be one chromatid, this one will be another chromatid. So bivalent chromosomes are separated into two separate different units in that of metaphase 1 of in anaphase 1 of meiosis 1. Two, that is homologous chromosome divided into two separate units. Here that is chromosomes. One chromosomes will be divided into two separate parts. That is chromatids. Okay, separation of chromatid. That is chromosomes. One chromosome will be divided into two separate units. That means two separate chromatids. That is the objective of. So you mark it that. That means how it will have to be happened. Okay, you see that. That means you see that. Suppose here is that of centrioles. So you can have to, that is separation part that has been joined. It is another separation part. This has been joined. This is another separation part. This has been joined. Okay, and this one is another separation part. This one is also joined. And this one, that is separated part. This one also separated part. This one also separated. This one also like this. So like this way, that is chromosomes divided into two units. One chromosomes become, it is that of chromatid. This is the chromatid part. Okay, this is the chromatid. You see that from homologous chromosomes to chromosome in anaphase 1. In anaphase 2, that chromosome segregated, that is divided into that of two units. That is chromatid. This one is the chromatid. Okay, if you have to draw it in a double manner. Okay, if you have to draw it in that of a double manner, because there are two nucleus are there, so you see that. That is, you are drawing here, that is anaphase. That is anaphase 2. You see that. That means here is that of one centriole, another centriole here. Here is a one centriole, that is another one. Okay, so from one nucleus, that means they have been separated. You see that. That means this is separated. This has been separated. Okay, another separations also occur here like this. So that means it will pull to that side. Okay, you see that. You see that. Mark it that. That means this. That is spindle fiber that is pulling to that line. That means they are being pulled towards that is polar. Okay, pole, that is to that of polar regions. And this one also being pulled to that in opposite direction, that also being drawn. So that it is happened in that of one nucleus. Same thing also happened in that of, same thing you mark that. That is same thing also happens in that of another nucleus. You see that. In another nucleus, that is same thing happens. Okay, same thing happens. You mark it that. That means in one nucleus, side one nucleus. In one nucleus, you see that so many, that is these chromosomes divided into two units. These are the separated chromatids. These are the separated chromatids of one chromosome. This one also, that is separated units of chromosomes. So this is a one nucleus. This is another nucleus. Okay, that things happen in anaphase. You see that, and that has been made that of four groups. You see that. That means one pole that is has drawn that pulled all these chromatid towards it. And here also that of in anaphase, that means that have been drawn to that up. So here is 1, 2, 3, 4. Okay, that means in mark that in one nucleus that has been two parts. It has been two parts. One, that means these chromatids have been drawn to that of a centriole region, that is polar region. These chromatids drawn to that like this way also, that have been pulled to that of a polar region. So that means in that 1, 2, 3, 4. So that 4 types, you should mark it that is 4. You can have to see that there are 4 regions. Here 4 regions have been created in anaphase 2. That means separation of chromosomes into 2 units. So as there are chromosomes in nucleus, they have been separated and formed that of 4 units. Okay, 4 units. And then you see that then there will be sister chromatid. So that is the separation of that of a sister chromatid. So separation of a sister chromatid happens in that of a anaphase 2. Then we'll have to go to that of a telophase. Okay, in that of telophase, you mark that. That means in telophase, I'm just drawing here. Again, I will have to draw. It is called that of anaphase. That is anaphase 2. Here is that of telophase 2. Okay, telophase 2. You mark that. That is one centriole. That is one centriole. Okay. Here is that of here that of what? These are that of sister chromatids. Okay. Here is that of sister chromatid. They have been separated. After that, they are going to that of polar regions. These are also okay. You see that. These are also they are joined together, and you see that. That means in anaphase 2, that sister chromatids are being separated. Okay, how sister chromatids are being separated in that of anaphase. Then they will go to that of a telophase. In that of telophase, what will happen that students, it is very important that means we see that all these chromatids will go to that of in that shell. In that of cell, how cell will be divided into that of 4 parts. You can see that. That means this chromatid, this sister chromatid will go to that of 1 unit. Okay, that is one nucleus. That means one nucleus will be formed here. Another nucleus will form here. You see that these chromatids join to that of nucleus, so that is one nucleus formed. Then another nucleus formed. There are one nucleus here. They become 2. This one also one nucleus. They also become 2. So in this way, they will be divided into that of 4 cells. So in meiosis, after meiosis 2, what you will have to get? That means after meiosis 2, there will be one cell. That is, you see that in meiosis 1 cell becomes 2. In meiosis 2, that means how 4 cells are produced. Okay, how 4 cells are formed in this way, and they will be, that means these chromatids will be that will be chromatids. Okay, that will be chromatids. They will be compacted and they will be formed that of chromatids. This will also that of chromatids. So here we will have to find that of chromatids. Okay, chromatids that will be formed of that of chromatids. Okay, so that you see that this one becomes and this one become this one. So this one also become this one also become this one. So you see that. That means how in telophase that 4 nuclei are formed. Okay, 4 nuclei are formed. So after cytokinesis, then there will be cytokinesis that will be divided into 4 parts. You see that. That means first of all, they will be divided like this way. First of all, they will be divided like this way. This is called fast kinesis. Fast cytokinesis. This is called fast cytokinesis. They will be divided into 2 parts. Here 1, 2 like this. So that here then they will separate like this. This is called second kinesis. Okay, cytokinesis. Then they will be, so that will be 2. So that is 2 cytokinesis. Then there will be third cytokinesis. Okay, so there will be 3 cytokinesis after telophase. The first one is like this. that is called cytokine. First cytokinase, that will be divided like this way. Okay, on that like this way, they will be divided into two parts. Then this part will be segregated into two units, two shell. Then this is the second cytokinases. After that, this part will be divided into two units. That is called that of third cytokinases. So there will be three cytokinases takes place. So this one is called that. This type of cytokinases, students, is called that of one is called that of continuous cytokinases. Okay, this one is called that of continuous cytokinases. That is successive cytokinases. One is called that of successive cytokinases. Another one is called that of simultaneous cytokinases. In successive cytokinases, you see that this is the one cell. Okay, that cell after meiosis, you see that after meiosis, it is divided into that of two shells. So that you see that is called meiosis one. Then after meiosis, you mark that. After meiosis, you can have to find that. Okay, after meiosis, you can have to find that of that is what? That is formation. So that is meiosis 2. So meiosis 2, and after that you will have to find that there are 4 cells formed. So cytokinases here, one cyto, that is called successive cytokinases or succession. That is meiosis 1, then 2 cell form, then meiosis 2. After that cytokinases, 4 cell form. That is cytoplasm divided. So 4 cell form. That is called successive cytokinases. But this one is called simultaneous kinases. That means it will divided first like this way vertically, then it will have to divided like this way. So this is the first cytokinases, this one second, this one third. So this is called simultaneous cytokinases. Okay, there are two type of cytokinases. One is successive cytokinases, in which that is there will be a succession. There is a order. Okay, that is meiosis 1, then meiosis 2, then cytokinases happen. But in simultaneous cytokinases, you can have to find that is meiosis 1, that is meiosis 2, that is meiosis 2, then meiosis 1, that is then meiosis 2, that is 4 cells will be formed. Then after that cytokinases, then there will be 4 cell forms. Okay, that is called simultaneous cytokinases. So there are two type of cytokinases, that is successive cytokinases. Another one is simultaneous cytokinases. In simultaneous cytokinases, there are number is cytokinases number is 3. But in successive cytokinases number is 1. Okay, that is the basic difference. And that of importance of meiosis. That is important of meiosis is, students, that it makes that of genetic variation. Okay, genetic variation. Mitosis, in mitotic cell division, there is no genetic variation. Okay, there is no genetic variation. All are alike and similar. But in that of meiosis, you can have to find the genetic variation. So meiosis first significance is that it brings the genetic variation, dissimilarities. Okay, there is a dissimilarities what you can have to find, that of appearance or that of looking. It is due to that of that is genetic variation. That is due to that of meiosis. Okay, and meiosis, that is it is found in that of reproducing, that is sexually reproducing organism. It is found in that of diploid cells, but it produces that of that is gametes. That gametes are that of what? These are that of that is gametes haploids, and that of after combination of fusion of gametes, that gametes also become diploid. Okay, so that haploid turns into that of diploid. It is due to that of that is what? That is meiosis. Okay, so meiosis is generally found in that of sex cells and sexually reproducing organisms, and it divides one cell into that of 4 cells. Okay, 4 cells. And this is all about that of meiosis. Okay, so in meiosis, what you have learned that it is generally found in that of sexually reproducing organisms, and it has made the cells that is haploid, but after fusion it also come back reported to that of a diploid. Haploid to diploid is that main objective of that of meiosis, and it brings that of a genetic variation. Okay, in that of some organism also, when that of in anaphase space, you can have to find that. That means suppose there are 23 pairs. 23 pairs. In anaphase 1, what you will have to find here? That means 23 number of chromosome here 23, here also 23. But due to that of independent assortment, sometimes that means the number of pairs goes to upper, that is 24, and comes to down, that is 22. Okay, then it is that of a gamete. It is suppose it is found in male gamete. In female gamete, that is 23 pair, 23 pair. So 24 plus 23 will be 47. That means in sometimes it is also found that is called aneuploidy. In aneuploidy, what you will have to find? Number of chromosomes decrease or increase. Increase and decrease of number of chromosomes in a cell, that is called aneuploidy. That is called aneuploidy. Aneuploidy. Okay, that is ploidy. That is aneuploidy. P-L-O-I-D-Y. Aneuploidy. A-N-E-U. Aneuploidy. P-L-O-I-D-Y. Ploidy. Aneuploidy. That is, you see that. That is, suppose male gamete has 24 chromosomes. Here 24. Here, that is in female gamete 23. After fusion, there will be 47. That is chromosomes. Here down part is 22. So 23 mix. Here will be 45. So there will be increase or decrease. Okay, that will have to find in that of that is Turner syndrome, Klinefelter syndrome. Next year we will have to read in the next, that is in 12th class, you will have to read about that of aneuploidy. But little bit I have remembered, come to my mind, so I have told it. That is aneuploidy. That is also found in anaphase 1. Aneuploidy and that of law of independent adjustment is found basically in that of that is anaphase 1. Okay. So this is all about that of meiosis. I have finished this chapter, that is cell cycle and cell divisions, and from that of next class onward, we'll have to start with that of human physiology. Okay, human physiology, that is 7 chapters. This 7 chapter completely, and you will have to finish one by one, and it will take that of nearly 28 days. Okay, and that by the end of February, we will have to finish that of human physiology. Okay, students. Now that is only one question I will have to do today, and so that after that we will go for lunch. Okay, that is one question I am doing, and you cooperate to me. That question is that very easy questions I am making, that is match the columns. Okay, that is match the columns. You must do it. That is match the columns. You see that column 1 and column 2. Question number 1, that is match the column. Match the column. That is column 1 and another one is column 2. Okay. So you see that. That is A is, that is leptotin. Okay, leptotin. 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 That is B is zygotin, C is pacritin, D is diplotin, and E is dikinesis. Okay, kinesis. Very mindfully you see it. Chiasmata I. Chiasmata. Okay, chiasmata. Then come to that of synoptimal complex, that is synoptimal complex. Okay, and come to that of 3, that is compaction of chromosomes. That is compaction of chromosomes. That is IV, that is condensation. Condensation of chromosomes. Condensation of chromosomes, fully condensed chromosomes, that is condensation, full condensation of chromosomes. And the last one is that of recombination modules. Recombination modules. Okay, students, I'm giving you options A, B, C, D, E. Okay, I'm just writing option. You also write with me. That I, triple I, V. Okay, then triple I, B. Triple I, V. Okay, V, that is V, V. Okay, V, then V, that is triple I. And the D is that of triple I, V, I. Okay, students, now you give that of answer. Answer is yes, Bhagavat Hembram, right answer. Smaranika, no, Smaranika, you are wrong. Bhagavat Hembram has the right answer. Brilliant answer by Bhagavat Hembram. Okay, Sanbhi Parija, that is Mona Lisa, no, wrong. Smaranika Patra, wrong answer. Bhagavat Hembram, Mona Lisa Rautrai, then correct, that is B. Okay. And Biswajit Mahalik B, right answer, Biswajit. Okay, Biswajit, Bhagavat has given the right answer first. That Sanbhi, you just show your answer. Sanbhi, you just show your answer. That option, you just show me that A, B, C, D. Which one is correct?
Sir, I have sent B twice.
Okay, you have sent B twice. So the B is the right answer. Okay, that B is the right answer. So you see that leptotin, I'm just drawing it. Leptotin, that is compaction of chromosome. Compaction of chromosome happens in leptotin. Then come to zygotin. In zygotin, you can have to find that of synaptinimal complex. That is synapses form. So synaptinimal complex, that protein joins, that is two chromosomes together. So that is called synaptinimal complex form in zygotin. So that one will be II. Then pachitin. In pachitin, what happens, students? That in pachitin, that is happens that of B. That is recombination nodules. In pachitin, recombination nodules you can find. In that of diplotin, that is in that of diplotin, what you will have to find? Chiasmata. Okay, that is chiasmata. And in that of dikinesis, that is full condensation of chromosomes. So answer is B. Okay, so all the students that has been sent that of that Bhagavat Hembrams, Vishwajit Mahalik, Sanbhi Parija, Monalisa Rautrai, very brilliant students have answered correctly. Good answer, brilliant answer. Many thanks to you. You have understood that person. That is very important that you have understood, and such type of things will come in examination, so that you have to remember. Thank you, students. From next day onward, you'll have to do more questions. Okay. So thank you very much. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you.